This is TWIS. This Week in Science, episode number 834, recorded on Wednesday, July 21st, 2021. Fly me to the moon, Mr. Bezos. Hey there, I'm Dr. Kiki, and tonight on the show, we will fill your head with butterflies, ice, and assimilation. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Space, the final frontier. A place where one day mankind will watch as billionaires build resorts on the moon and overtime regulation-free factories on Mars. Space, the undiscovered country from whose bourne no travelers' tax returns are ever seen. And while tax-dodging billionaires live out childhood dreams of wearing spacesuits and launching their rockets into suborbital altitudes, and then fizzing all over themselves with champagne upon completion, let's not forget that this is a great achievement for the commercialization of space, and a rather modest admission that as a nation, America will play no role in it. When NASA landed on the moon, we did so as a nation, and the words Neil Armstrong used dedicated the event to all mankind. Jeff Bezos made no such statement. Well, he did thank Amazon employees for paying for it. I suppose through starvation wages, unsafe working conditions, unrealistic time quotas, lack of job security, lack of health security, and an all-around lack of respect for basic human dignity. Never mind the unregulated outsourced manner in which most of the products the company sells are manufactured. Somehow, all those starvation wages equated to job well done. You did it. You put one billionaire space cowboy into suborbital something. Look around at the growing homelessness problems in fill-in-the-blank city or town. The housing epidemic, the lack of mental health programs, the growing expense of healthcare, the cost of educating the next generation, the stagnant spending on science, research, and the lack of activities to do with global warming. The rest. And tell yourself, hey, at least we are paying for billionaires to take publicity-fueled joyrides instead of collecting taxes for them. Job well done, everyone! Who needs public funding for society when you have that? The limited public funding of science is, however, the only reason that we can even bring you another episode of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. Brought to you by the funding of science. Yes, that's true. And our Patreon sponsors. But we have a whole episode full of amazing science ahead. And I guess, as everybody's here joining us for this episode, I hope that it will distract us from the fact that the billionaires are escaping to space while the Earth floods and burns. Little issues like that. On this week's show, I have stories about assimilation, about protein structure, AI, and also about helping your own with empathy and rewards in your brain. What do you have, Justin?
I've got a few words about how to fight global warming, a new species of butterfly discovered in San Francisco, why there's no room for new life on the ocean floor, and why 15,000-year-old novel viruses are being thawed from glaciers.
That sounds like a good idea.
Great idea. Wonderful idea.
What could go wrong?
So, and the fun byline on that one is, it's happening anyway.
Oh, right. Yes, that whole
right, yeah.
climate change thing.
Yeah.
we talked
It's
about.
happening because of the warming, and so why shouldn't we help? Blair, what's in the animal corner?
Oh, I have shark poop, I have Pika poop, and I have fish friends. There you go.
Okay. Fish, fun and poop in the animal corner. Okay, great.
Yeah.
You know what we like to hear from you, and you
It is, yeah.
definitely bring it.
Listen, I have a lane, and I am sticking to it.
At least I don't have to shout at you and ask you to stay in your lane, Blair. Everyone who is with us, thank you so much for joining us. If you have not yet subscribed to This Week in Science, you can find us all places podcasts are found. Look for This Week in Science. And you can find us on YouTube, Facebook, and Twitch. Look for This Week in Science. Our website is TWIS.org, TWIS.org. And now it is time for assimilation. The Borg.
Mm-hmm.
We're going to talk about the Borg.
Resistance, I have heard, might be futile.
Yeah, well, resistance against me talking about this particular story right now, totally futile because we're going to talk about it. And when I talk about the Borg, I'm talking about not the Star Trek civilization of robot people, cyborgs that assimilated any living being into their super entity. What I'm talking about are linear elements of DNA found outside of bacteria in mud. That's what I'm talking about. Some Berkeley researchers have found what they call ELEs, EGEs, extracellular genetic elements, these extracellular line elements. And sometimes these extracellular elements are circular in shape. The circular shape preserves the DNA, protects it while it's in the dangerous and inhospitable environment of water or mud or whatever environment it's in. But these elements are made of DNA. They are not inside bacteria per se, but they can be picked up by bacteria and then used and swapped around by bacteria. Wow. And this is very often how antibiotic resistance gets swapped around, are these extracellular elements. And they're usually very short. However, these particular elements were massive. They're like over a third of the size of a piece of a chromosome for archaebacteria. Archaebacteria also tend to have pretty large sizes, and they're these amazing survivors in the bacterial world. The protobacteria, bacteria that came before bacteria, that are extremophiles. And in this particular situation, the researchers dug a little deeper, did genetic analysis, and found a whole bunch of genetic elements within these, what they're calling Borgs, because of the ability of these extracellular elements to pick up DNA from other bacteria and help swap them around. They assimilate genetic elements very well. Yes, they do, these Borg. They looked deeply into the genes that were contained in them and found genes responsible for the metabolism of methane. And they found that these elements seem to be associated with methane-eating archaebacteria. And there are a number of questions still at play here. The researchers, number one, have not been able to culture these archaebacteria or the line elements in the lab, so they can't actually confirm that they're really associated and being used, associated with and being used by these archaebacteria. And because that's an issue, that it's kind of a question as to whether they really exist at all. I mean, they found them. They found the genes. It's a lot of gene sequencing, but they haven't been able to show proof in the lab at this point.
Okay. I mean,
and the study is also a preprint, so it hasn't been peer reviewed yet. But people are talking about it, so it's a very interesting question.
So is the idea that these are like lysed cells from archaeobacteria that then other bacteria are like, oh, I know how to use this. As long as they're not using this bit, I can incorporate it?
Yeah, so I don't know how they would end up, how these elements are just floating around in the mud. But apparently there's DNA all over the place, and it just gets picked up and swapped around by bacteria all the time. Not necessarily from lysed bacteria that have exploded and spewed their contents all over the place, although that's potentially how you would think that kind of an element would end up in the environment. Yeah, so random genetic elements found in the mud, researchers calling them Borg. They're really long, and they have these interesting aspects to them. They did discover that they have these repeating elements at the caps, at the ends of their length, which is unusual. It's very similar to stuff like CRISPR, where they have the repeating elements. These extracellular elements, the circular ones also very often have a lot of repeats contained within them, and they find that as well. But they're wondering if they can get the culturing to work, if they can potentially, not in the wild, in, like you were mentioning just a moment ago, the melting permafrost that is releasing methane, maybe not muck around with genetic elements and bacteria there, but they're going to start looking at rice fields in California to see if they can perhaps add in this extracellular element, these Borg, with these methane-eating genes to see if they can reduce the amount of methane that is produced in agricultural processes.
That's a fun question. My number one suspect of this is that there's a virus at play. If it is doing an insertion spot, capping things, like tearing up the cell, putting those exploding the cell, or having it otherwise be torn to shreds, then that opens up this whole question: like, how big and important would then viruses be in doing this process over all of evolution? Tearing up one thing in a way that something else could incorporate it.
Yeah.
They then become the workers for that. They got to look for the virus. There's going to be a virus in there that's tearing out these archaeobacteria, that's allowing them to remain stable and get incorporated later.
Yeah.
And then the thing about agriculture methane, that'd be fine too.
That would be fine too. Yeah, we need to reduce the amount of methane that we release into the atmosphere. That is a huge thing, especially if the permafrost is melting and speeding up the process of warming in the atmosphere. I don't know about any evidence about viruses. All they have are these long sequences of DNA that are hanging out in the mud, but that apparently exist in tandem with these methane eaters. I don't know. Yeah. Anyway, Justin, what did you want to talk about next?
Oh yeah, let's go to some viruses were found in two nearly 15,000-year-old ice samples taken from the Tibetan Plateau. Most of those viruses are unlike any viruses that have been cataloged to date, and because they were frozen, they survived. They're perfectly fine. Yeah, they're perfectly fine. The findings published in the journal Microbiome could help scientists understand how viruses have evolved over centuries. And evolved in viruses is tricky because they don't have this sort of reproduction that we normally constitute with something that's alive. And they do replicate, but it's different, right? So for the study, scientists created a new ultra-clean method of analyzing microbes and viruses in ice without contaminating it. These glaciers were formed gradually, along with dust and gases. Many, many, many viruses were deposited there, says the Jiping Zhang, the lead author of the study. They analyzed ice cores taken from this ice cap in Tibet. These cores are also at a very high altitude. The summit there is 22,000 feet above sea level. So this is a sort of interesting laboratory for a lot of different types of research, but it gives them a timeline of sorts going back over many millennia to look at things like climate change, microbes, viruses, and gases throughout history that have been trapped in the ice. Yeah, but they found genetic codes for 33 viruses. 4 of those viruses we have already discovered. 28 of them are novel. About half of them seem to have survived from the time that they were frozen but not just because of the ice. Not just because of the ice. These, Matthew Sullivan, who's a co-author of the study, professor of microbiology at Ohio State. These are viruses that would have thrived in extreme environments. These viruses have signatures of genes that help them infect cells in cold environments. Just surreal genetic signatures for how a virus is able to survive in extreme conditions. So part of what they've also done is they've used a new method that Xi Jinping has developed to decontaminate the cores and be able to even study the viruses that were contained within the ice. They now think they could actually use this technology on Mars, on the moon, and actually even in desolate areas where we can get to ice that hasn't really been being disturbed by mankind. As much of it as is existing now, anyway. So one of the things, too, that they're going to be able to study, this is according to Lonnie Thompson, who's a senior author of the study, as well, also at Ohio State. We know very little about viruses and microbes in these extreme environments and what is actually there. The documentation and understanding of that is extremely important. How do bacteria and viruses respond to climate change? What happens when we go from an ice age to a warm period like we're now in? So some of this will help our predictive ability for what's going on in the future. And yeah, as scary as it might sound that we have taken 28 novel viruses from an ice core, like we were teasing earlier, this is happening every time the ice melts. Mm-hmm. And we're melting a lot of it. So we're going to encounter this anyway. Also, when they looked at these viruses, they say they look a little bit more typically like a soil virus. Something that there are trillions and trillions of all around us, and most of them are going to be novel because we haven't looked at everything. Not necessarily because they're so frighteningly different. Mm-hmm. But one of the also problems with viruses is they do not have the type of evolution that we associate with life forms. So when we look at a virus, we can't easily determine what it's related to, what it's, especially with something that's maybe as disparate as 15,000 years of virus evolution or viral changes, mutation that has taken place over time. You can't simply look at a bit of that and go, oh, I can tell that it diverged from this virus, 2 million years ago, or how things that we can actually do with our life genetic code.
We can, though, scan the DNA that's there and look for genes that are very similar to what we see in modern viruses, what we see in other bacteria, and we can look at what we know to be certain genes with specific functions. And if, that may not tell us lineages where the virus potentially came from, but at least it can tell us what that virus could potentially do.
What its target is. You can sort of see what does it eat. And that tells you then a tremendous amount of information about what it was in the environment and when it was with 15,000 years ago, which really isn't that long ago.
It's not that long, but in virus lifespan, it's probably, that's like so long. Like we're seeing how fast SARS-CoV-2 is mutating, and if a virus has the potential to spread and mutate, it's going to, and it can change very rapidly. Yeah, depending on where it is and what it is doing. Beware of the Tibetan ice.
Oh, it's fine.
Yes, the permafrost is really what we should be more worried about. And I think that's I'm going to keep going back to that for the rest of the show. Nope, the permafrost. Mm-hmm. Beware of the permafrost. Blair, what do you have?
Well, you know who's not hiding in permafrost, I don't think?
Who?
Sharks.
Oh, yeah. Okay, I hope not. Yeah.
Yeah. So I actually have a study about shark intestines. Did you know? We don't actually know that much about
I'm just going to stop you right there. You said shark intestines, and you said, do you know? And I'm going to say no.
No.
I do not know.
You're right. No. And you are not in the minority there. So even people who study sharks for their whole lives don't fully understand the complete picture of what sharks eat and how they digest it. And that's because they've had to cut open shark specimens to figure out what's going on in 3D inside their bodies. So they either get that or they get flat sketches of sharks' digestive systems. So this is the first time that researchers have produced a series of high-resolution 3D scans of shark intestines from nearly 36 shark species, not individuals, but species. This is a pretty widespread study. And they're looking at something that apparently shark biologists did know, which is that sharks have spiral intestines. And so it looks like a corkscrew inside of a tube. There's really no other, it's a corkscrew spiral intestine. And this is a study coming from University of Washington, California State University, and University of California, Irvine. And CSU Dominguez Hills and UC Irvine. And they developed a new method to digitally scan these tissues so they can look at soft tissues in detail without having to open the shark up.
That's
they use a CT scanner, computerized tomography, and they did it just like how certain X-ray images can be taken, where you take them from different angles and you kind of stack them up with computer processing to create a three-dimensional image. So you're able to do that all from Natural History Museum specimens from the Natural History Museum of Los Angeles. And they found, based on this corkscrew kind of structure inside the intestines, that this spiral-shaped organ, it actually slows the movement of food, which makes sense that you could travel straight through, or if you have to go in a spiral, that's going to take longer. So they slow the movement of food, and they direct it towards the gut, so you can't really go backwards up. You can only go through. And that relies on gravity.
Once you're swallowed by a shark, there's no getting out.
Until you get out of, you could get out if you're in the stomach, but then, once you get to the intestines, that's it.
Why is there a Tesla valve on the image?
Great question. So it resembles the one-way valve designed by Nikola Tesla over a century ago that allows fluid to flow in one direction without backflow or any moving parts. So why is this important? So on one side, you have learning more about sharks and how they eat. One of the crazy things about the way sharks eat is that they can go weeks, they can go days to weeks, I've heard in cases months, without eating. That means they are able to hold on to food in their system to absorb many more nutrients than we would in our body. This could help explain that. You're increasing surface area, you're slowing it down. It's a one-way travel. So they've slowed the movement of food. But the other piece of this is, because it looks so similar to this valve created by Nikola Tesla, they actually think once they use a 3D printer to create models of these intestines, they could test how it works in sharks, but also work with engineers to use this model, a shark intestine model, as inspiration for industrial applications like wastewater treatment or filtering of microplastics out of the water column.
Wow. Thank you, sharks.
Right.
Yes. Thank you, Nikola Tesla, and thank you, sharks. Also, what was Nikola Tesla looking at? That's what I want to know. Did he know?
The intestines of sharks.
About shark intestines?
Right.
Maybe
he had a fascination we're not aware of.
It was a brilliant invention because he just took the properties of water pressure and created a one-way valve with no moving parts.
Exactly.
And it's the simplest structurally design to efficiently to do that, and so of course nature figured it out way before we did.
Exactly. Yeah, that's what I love about it, is whether he knew about these kind of structures in nature or not, he stumbled across something that nature figured out millions of years ago. Don't even worry about it.
Now I'd like to know if there are other species that have this kind of spiral intestine design, if sharks are unique.
Good question. And we'll be able to figure that out by using this new CT scan technology, or the technology is not that new, but the application is pretty new. So anyway,
that's really neat. And speaking of cool applications of technology, and I mean, it's not an adaptation, but it's an improvement on artificial intelligence. We've reported previously on the effort by Google's DeepMind AlphaFold to determine the structure of proteins. And this is something that we've talked about for years on the show, going back to Fold It, a game that uses human pattern recognition to be able to suss out the patterns and the folds and the twists and the whirls in protein structures. Now, and if you remember, a little over a year ago at the AAAS meeting, we did a live show and we interviewed Dr. Baker from the University of Washington Institute of Protein Design, and we talked with him about his synthetic biology. Well, this new development is out of his lab, the Baker Lab at the University of Washington. They have developed, based on what was revealed at the meeting that we talked about like six or 8 months ago, that Google Mind's AlphaFold project did like 90% accuracy in determining protein structure. The stuff that was revealed about their methods and how they did it, the Baker Lab and their collaborators, they used that information to make an open source artificial intelligence, because Google DeepMind's AlphaFold is not open source. They have made an open source AI for determining protein structure that takes only 10 minutes to determine the structure of a protein, running on a gaming computer.
Wow.
Yes. So they're using, off the shelf gaming. It's really good gaming computer processors, but at the same time, this is off the shelf technology, and they've created this artificial intelligence using very basic rules that are on par, almost as good as in the 80-85% accuracy range, which is above all of the other artificial intelligences that are out there working on the same problem. So it's really starting to get at this problem of protein folding, and in a really rapid way. Single pass, 10 minutes. Oh, look, here's your protein. You may have to do confirmations, but the fact that it's open source, will allow real development to take place from here. Yeah. Artificial intelligence. Yeah, what do you have?
Speed at which this stuff is happening is just exciting. What have I got? Oh, I have an editorial published in Energy and Environmental Materials. Professor Ravi Silva, director of the Advanced Technology Institute at the University of Surrey, is arguing that there are no coherent excuses left to justify the use of fossil fuels. Such a great way to put it. No coherent excuses, which does leave
but society, but economy, but yeah, no excuses.
Plenty of room for incoherent excuses. So he's, in his paper, Professor Silva challenges the scientific community to lead the world away from fossil fuels, which are still 80% of the energy mix at the time. While the cost of clean energy generation has been plummeting, it's going down. The cost of getting that energy is cheaper and cheaper. Silva argues that significant innovations in advanced batteries and energy storage technologies are needed to keep up. They are needed to meet the International Energy Agency's goal of a planet being carbon net zero by 2050. For example, as pointed out here, the transportation sector would need to see a 15-fold rise in electric vehicle sales from 10 million in 2020, which is still a really good number, to 145 million in 2030. And that's not an unachievable goal by any means. So but it is dependent on a leap in some battery and energy storage technology, as well as, which isn't pointed out here, the ability to charge anywhere. Which I was actually just excited. There's this oldie time bank downtown, small town Davis. And I just drove by it for the first time in I don't know how many years. And their parking lot has charging stations in it now. Which is one of those things that this innovation can be done within our existing infrastructure. It's not like we have to build a different kind of gas station next to the old gas station or on top of it, or we can put this is something that's very accessible. This is something we could put in parking spots pretty much anywhere. Let's see. So the quote here. Well, first he concludes that the unprecedented but much needed goals are only possible if the scientific community itself ushers in the new wave of energy materials that are cheap, easily deployable, and have short payback times. This is quotey voice of Silva. The pandemic has been a truly horrific experience. However, one of the few positives that I can gather from the past 2 years is that it has allowed me to take stock and refocus on the incredible challenge of combating climate change. It's increasingly clear that the energy materials community has a crucial role to play in weaning the world off of fossil fuels. The cost of green energy is falling all the time. In the UK, solar and wind generation is competitive with fossil fuels, but we need to look at improvements in thin film technologies, new polymers, and other hybrid materials that can boost energy capture capabilities while reducing the cost of production if we are to have a genuine green energy revolution. So calling out it sounds like very much his peers and the companies that are involved to refocus some of their energies and resources towards the materials that we need to make these next big steps.
Yeah, one of the things that we do need to consider for these big steps, there is a lot already available, like he is arguing and like you're saying, but there are big questions about the sourcing, the ethical and sustainable sourcing of materials like lithium for car batteries, or how do we do that without destroying the oceans? How do we get this material that we so rely on, and how do we do that in a way that doesn't put the burden unduly on underrepresented groups around the world, on the more impoverished groups around the world.
Such an important part of this, right? Is like, it costs money. It costs money. Even if it's cheaper to have the green thing, if you're replacing a thing you already own with a green version, it costs money to do that. And so unless governments are willing to help with that process, it's going to be really difficult logistically to do this without disenfranchising people in lower incomes.
Yeah, I disagree. Okay, so like this is why when the first generation of Prius came out, they had actually cost something like $10 million each to produce, if you factor in the R&D and everything else. A few years later, there's 30 million of them on the road, and they're the same cost as a regular compact sedan. So that hurdle is sometimes an illusion. Like, we're looking at Nikola Tesla's doing these high-end electric cars. That need not be the electric car that every man drives, and it need not come at those price tags.
Right,
so
but we've also seen different countries and states put forward assistance. So there are, you can get rebates for buying an electric car or a hybrid car, and there is assistance that has allowed the spread of these vehicles to happen more rapidly. There's also assistance to actually help the companies make the transition from creating gas cars to electric vehicles. It's like, oh, if you're going to sell cars in our state, then you have to have at least one electric vehicle that you sell among all your other cars. So there are regulations that push that development forward. And, it's hard to get past that $100 million prototype into mainstream acceptance and distribution.
And I mean, ultimately, so I just bought a car 3 years ago, right? And I was planning on driving that car until it died. The last car I bought, I drove for 15 years, right? But if I am going to hand in my car for an electric vehicle, even if it is reasonably priced, it takes a certain amount of financial privilege to do that before I was planning to do it. And that is the part I think that gets missed here. Is it's very easy to say all new buildings are going to have solar panels. But what about people that have owned the same house for however many decades, right? And so there has to be a conversation, not just about making sure new stuff is better, but about making sure that there is a subsidized practice of trading out for newer stuff. And there are things like washing machines, like you can get assistance to trade out an old washing machine or an old refrigerator to get a new one that is more energy efficient, but it's not no dollars, which is what it takes to keep the one you have.
Yeah.
Well, I that makes some sense. There's detractors to that as well, as the cost of maintaining a vehicle is totally different on an electric car. Like, you do away with repairs. For the most part, you do away
right, but
with
it takes a big
and
cost all at once. And if you're poor, you can only pay for the small repairs in little bits.
Sure.
But then there's also the resources themselves, and so like pipelines going through indigenous lands, there are going to be those questions about where is the lithium going to be mined for these batteries? How are we going to get it? Are we going to just say, oh, it's on Native American land, so whatever? You know, we have to put forward sustainable and egalitarian practices moving forward if we want to change the way that everything works.
And I love that idea. I think that might have to come second, unfortunately. So, and what we were talking about, to be fair, is we're talking about the burden being the cost of these things, environmentally or what have you, being borne by certain different people. It's different underprivileged people than the underprivileged people who are burdened by pollution in inner city and fossil fuel factories. And that's still, I mean, it's not like we're adding that. I want to make that clear. We're not adding
we're just shifting the burden. So that's fine. It's like move the deck chairs around. We need to remove burden is what needs to happen. That's what I'm arguing.
If the point is that we're severely and drastically reducing it for all ships, we may be able to find some ships who are more negatively impacted, but I don't think that when we're lowering that burden on all ships together, I don't think we have to make that the thing that stops. Right. Giving up the perfect for the good or whatever
we still need to push forward, but exactly. I think we all agree on that. Yeah. And the editorial, what you're reading, absolutely agree. And we need to make sure that it happens. But there are better ways to do things. And that's only going to happen if people push for it. And my final story for this opening segment is that we've talked about spider silk on this show many, many times. Researchers have been trying to create synthetic spider silk for years and years and years. Well, researcher publishing in the journal ACS Nano from the Department of Energy, Environmental, and Chemical Engineering in the McKelvey School of Engineering at Washington University in St. Louis has created bacteria that are engineered to make spider silk that's stronger than silk that some spiders make.
Stronger than spider silk?
Yes, this is synthetic spider silk that is stronger than spider silk. And they were able to, in previous studies, determine some of the compounds that were necessary to the tensile strength of spider silk. And they were able to solve some of the problems of mixing the various materials together that spiders have evolved to do so seamlessly. And in doing this, have managed to out-spider silk the spiders. And the researchers think that they can even do better with a little bit more work. That the researcher says that there are many more amino acids to be looked into. And there are many more combinations of chemistry to see. And so they've got their work cut out for them. But what this does suggest is that we are figuring out how to make silk fibers that are stronger than steel, yet flexible. And I mean, seriously, Spider-Man is coming. Except maybe, you'd have to have a better shoulder, maybe, to be able to swing at the bottom of the swing. Anyway, yeah.
You have to look a lot more ape-like, yeah.
I was picturing a spider silk bridge, and how weird it would be to drive on a suspension bridge. Go, it's made out of spider silk.
Exactly. Maybe, suspension bridges not with steel cables, but with spider silk.
And my brain is telling
lightness.
me, heck no. But it's stronger than steel.
I just want underwear elastic bands that can last as long as I'm willing to keep underwear.
Maybe one day with this technology, you will.
Yeah. Because that's the thing that always goes wrong. Anyway.
I don't know if it would have that much give, but...
I might be worried about... Yeah, okay. We got to talk a little bit more after the show about the underwear issues here. But for those of you out there, thank you for joining us for this episode of This Week in Science. We are back this week. Every week, we broadcast weekly on Wednesdays at 8 p.m. Pacific time, live on our YouTube, Facebook, and Twitch channels. If you enjoy the show, tell a friend. I'm going to come on back with a very quick COVID update. I've got a few stories to dive into, but they're fairly just not a lot of nuance, just a little bit of information. So I'm going to tell you what you need to know. COVID update! ... People who are vaccine hesitant sometimes say that they don't want to get the vaccine until it is fully authorized by the FDA. And so the question is, when will the COVID vaccines be approved by the FDA? Well, Pfizer has applied for its mRNA vaccine to be approved, and they are currently getting all the paperwork together to submit to the FDA. It has been stated that with the priority review process, which can take up to 10 months, that they set a determination date, and that date could be as late as January 2022. However, there was a report from an individual on CNN recently suggesting that it might be within the next couple of months. So we may see full FDA authorization for Pfizer's vaccine very soon. And then, as for the question about pediatric vaccines, the under 12s, what is going on there? They are currently running trials with reduced doses of vaccine for the shots that the children get. They are collecting data on those. They are in a second round of trials at the moment, and we could see an emergency use authorization for kids under 12, at least to about the 5th grade level. From 12, I think it's from 6 to 12. The under 6s may have to wait a little bit longer, but for kids of school age, we could see a authorization as early as September or October. They are rushing, but not rushing. They're doing everything to the letter, trying to get things done as quickly as possible. So we may see a vaccine for school age kids by the early fall. A little late for the beginning of school for some, but at least coming on the sooner side. However, there are also reports that this authorization may not come for all of the under 12s until at least midwinter. So probably December-ish of this year.
It's like a death wish. We keep not getting to the... before school. That's your deadline. Before school.
Well, we want to get the studies done to make sure that they have the dosage right, that things are as safe as can possibly be to reduce the risk, because the reason we want our kids to get vaccinated is to protect them, not just for the heck of it. So we need to make sure that everything is done well. Anyway, speaking of for the heck of it, if we've got about 70% of the population in a lot of places around the United States with at least the first shots of their mRNA vaccines. A study out this last week suggests that the second shot is really the money shot. That's the one that boosts, and it's a booster shot for a reason. It boosts your immune system, improves the T-cell response, and really gets your immune system primed to ward off SARS-CoV-2, much more significantly than just the first dose. So, if you've only had the first dose, really consider getting that second. it'll help your immune system fight SARS-CoV-2 off. And then the longer that this whole pandemic's going on and we're living with this virus, the more we learn about the virus and about our immune responses to it. You know, back this time last year, we only had about 6 months of experience with the virus. We didn't know what was going on. Now we've got 18 months of experience. We have people who have had the virus and who have survived and lived for at least 12 months, if not longer, with the virus. So we're getting more and more information. Another study that just came out this week found, it's in Nature Communications, found that people naturally infected with SARS-CoV-2 produce antibodies against the virus for up to 9 months following primary infection. And this is really great news.
Then they need to go get it again. Because for their booster, they need to go.
Well, so they also found that some individuals had increases in antibody production, and they think that that's the result of reinfection. So yes, people have gotten reinfected, and that also boosts the immune response. But yes, getting a vaccine is also an easier way to go through the process of gaining immunity without getting COVID, because you never know whether you're going to get stuck with those long-term effects, right? And then there's questions about kids and whether or not mild and asymptomatic cases actually lead to much immunity. They just released a study suggesting that children who presented with mild or even who were asymptomatic cases of COVID-19 had a really robust antibody response months later. Up to 4 months later is how long they followed for the study. So even a little infection for kids can help protect them. Although not saying you want to have COVID parties. That's a very poor idea.
No.
Again, when the pediatric vaccine comes out. COVID. Yeah. All of this is the great news that our immunity seems to last for. What we're finding is it seems to last, and that is really great news. The really bad news that I'm going to end the COVID segment on is that we've talked a lot about the adults getting COVID. We worry about about kids getting COVID, but then we don't really talk about the kids that are left behind after their caregivers die from COVID. And a recent study looked at the deaths of caregivers and the numbers of orphans around the world. There is over 1.5 million orphans from COVID, and an estimate at this point is that a child loses a caregiver to COVID once every 12 seconds. So you know what? Get vaccinated for the kids.
Yeah, if you won't do it for yourself, do it for your children so they're not alone.
Yep. Parents, grandparents, aunts, uncles. Just think about that. I think this was one of the hardest studies to look at and to read through this week because, you don't want to leave the kids alone and behind and as orphans. So that was some serious ending of the COVID corner. COVID update. This is This Week in Science. Thank you so much for joining us for another episode. We hope that you enjoy the science that we bring you, the good, the bad, the serious, the joyful, the fun, the funny, all of it. Because we like to talk about it with you. And you are the reason that we're here every week talking about science and trying to bring new discoveries to your ears. If you appreciate This Week in Science, please help us continue to do what we do every week by becoming a Patreon sponsor. Head to TWIS.org and click on the Patreon link to choose your level of support. $10 and up, and we will thank you by name at the end of the show. Thank you for your support. We really can't do this without you. And now we come on back to Blair's Animal Corner with someone, I think, named Blair?
She loves our creatures, great and small. Biped, millipede, no pet at all.
You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's animal.
What you got, Blair?
I have Pikas or Pikas, depending on what you want to call them. I call them Pikas. Pikas, Pikas. They are, I do believe, the inspiration for Pikachu, but they're actually real animals. They're mountain lagomorphs. Whoa, let me try that again. They're mountain lagomorphs. Lagomorphs are rabbits and hares, but they're also
into werewolves at night. Wait, what are they?
Rabbits, hares, and Pikas or Pikas. So they are a distinct group of mammals. They are not rodents. So people often think rabbits are rodents, but they're not. They're in this other group called Lagomorpha. And so Pikas, why am I talking to you about them today? Well, a University of Aberdeen School of Biological Sciences and Chinese Academy of Sciences study have completed a 13-year-long investigation on the survival strategies of the wow, I'm having a hard time. The plateau pika lives in the Qinghai Tibetan Plateau in China. They are of interest because they can't hibernate, but it gets really cold and all of their food disappears in the winter. How does this animal survive? Well, to figure this out, they went to the Qinghai Tibetan Plateau, around 4,000 meters in elevation, and they looked at the low temperatures and low oxygen that is present up there, or is not present, as the case may be, to provide, to look at how this specific environment still manages to provide for this animal and they still survive. So what they did is they measured daily energy expenditures of two different populations of these guys and found that the energy expenditures are about 30% lower in the winter. So they're spending less energy as they're running around. They also filmed their activities. They implanted them with temperature logging devices, and they also looked at what they were eating. So they looked at their stomach contents. They looked in essentially in their poop and did some DNA analysis. They found yak DNA in their poop. So obviously, they were eating yaks, right? That's obviously what. No. They're still tiny.
But that's fine if there's
They're nibbling
enough
on
of
the
them.
little yaks.
If there's enough of them hunting as a pack, they can jink down a yak.
Just a horde of pikas just coming up over onto a yak. Just all that's left is bone.
That's just destroyed by cuteness. Oh my goodness.
I think it's better. Anyway, that's not what they think, of course. They think that they are eating yak poop. Oh. This is especially interesting because in Tibet, yak herders think that pikas are pests because they think they're competing with their yaks for food in the winter when the food is less and dry and hard to find. So this would actually lead us to believe that they're around the yaks not to steal their food, but to eat their poop, which is a really important thing to then communicate to the Tibetan yak farmers, because if you explain that to them, maybe they won't be trying to intentionally rid their area of these animals that will be pressured by climate change, and they need all the space that they can get, right? So all that to say, they're eating yak poop. This is how they're avoiding the lack of food and avoiding hibernation. But the other really interesting thing is that the temperature logging devices on these animals also showed that their body temperatures dropped. So hibernation is essentially a drop in body temperature. It's a drop in metabolic rate, which is a drop in body temperature. And so they're sleeping. They're also kind of in a hyperthermic coma, essentially. That's kind of what it is, right? Is their temperature has dropped so much, their metabolism has slowed, but their body has adapted to allow them to do that without core systems failing. So these guys can't do that. They don't do that. They are able to eat, but they also lower their energy and they lower their temperature. So this is important to know also in relation to climate change. as things get hotter, will there be more grasses, which will then allow them to be more energetic and not lower their temperature as much? Or will the higher temperatures key their body to not drop? Their energy requirements will not deplete, but the grass will still deplete. This is also an option.
I think that's the interesting question, is what is it that triggers the change in metabolism? Is it just a change in day length? Is it a change in temperatures generally, where there maybe is a threshold temperature that when it hits a certain temperature for enough days that they're like, okay, that's it, my metabolism is going to be downregulated? Or is it food availability?
Is it food availability? Is it the type of food that they're eating? Is there a chemical change in the grass that they eat as the season progresses? There's lots of things that could be going on. But what's interesting is they have identified that lowering temperature, reducing energy output, kind of how much they're doing, and eating yak poop, those three things together increases chance of survival in the winter by 94 to 96% in these populations.
That's pretty solid.
So these animals have adapted to a very specific environment on the top of a mountain. So as I often talked about when I used to teach school programs about climate change, mountains, they're generally cone-shaped. And even though this is a plateau, it's like you took a bite out of the end of the ice cream cone. It's nice and flat on top. There's still nowhere further to go. So if they wanted to follow the colder temperatures that they're used to, they're stuck. That's it. They can't go down the mountain and up another mountain. They'd have to go through several biomes to do that, right? So this is just, they're stuck. That's as far in elevation as they can go.
Yeah.
So
they're on an island.
Yeah, exactly. 100%. Yeah. So it's little
and what
Pikas
happens when that island becomes inhospitable?
Indeed. Can they adapt quickly enough?
Poop-eating pikas?
Yes. And from pikas to fish, the tops of mountains to the bottom of the ocean, I wanted to talk about fish friends. So we often talk about animals and their relationships with one another within their species. They've developed friendships, associations, social structures, whatever fancy term or label you want to put on it. They have relationships. And when we think about long-lasting bonds or social familiarity, we talk about chimps, we talk about dolphins, sometimes we talk about rats, but we definitely talk about animals that we consider kind of like, quote unquote, like higher cognitive function. And so this is going to be one of those stories. Like, actually, all animals do it. So in this one, looking at fish and the importance of friendship to survival, this is a study from Nova Southeastern University's Holmes College of Arts and Sciences. And they wanted to study the presence of friends versus strangers in fish and how that changed their response to predators or a threat. So they took a bunch of tropical fish and they wanted to see how they responded to potential predators. So they had they had strangers in some tanks, and then they had friends in other tanks, fish that were very comfortable and aware of each other. They weren't strangers. And they found that the presence of strangers was actually distracting and made them react more slowly than the friendship tank when there was a predator. So the social scene affected their reaction time to potential dangers. Then they think that this comes down to trust. So as social animals in the wild, they rely on friends, buddies, comrades to alert them if a predator is lurking. So you can kind of let your guard down a little bit because you know there's other eyes on the field. You would think that would reduce their predator response time. Aha. But no. The issue is that if they are spending every minute looking in every direction, looking for a threat, it's harder to catch important information right away. So it kind of dulls their sensitivity to new stimulus, I guess. So the idea is you're chill, you're calm, you know your buddy next to you will notice a predator if they come. So if something unfamiliar shows up, they're like, uh-oh, what's that over there? But if they're around a bunch of strangers, they're already on edge. It's like, what's he doing? What's he doing? What's she doing over there? Are these fish my friends? Oh, I've been eaten. So the individual or the level of familiarity in a group impacts their ability to respond to threats. So this is the idea of the transfer of information, and if it breaks down, so does the chance of survival for the individual as well as the whole. So that means your readiness to react to a threat depends on who's around you, and therefore your entire social structure's ability to survive is dependent on familiarity. So this means there is a evolutionary advantage to developing familiarity, schooling, exactly. Yeah. Developing familiarity and developing relationships to delineate between strangers and friends.
Wow. We just think of fish in their big schools as just, they're just fish, right? But they know each other, they recognize each other, and it makes a difference to their survival.
Absolutely. Think about ants. Think about their social spiders out there. Think about I think there's a lot of situations where we could find more and more examples of this, where familiarity is important. And I think for people that work with wild animals, this was something that I think I wasn't expecting when I started working with animals, is that animals that you'd never think would care about you start to recognize you. And even if they're not affectionate towards you, they definitely recognize familiar from unfamiliar individuals. And that can't be just across to their keeper or somebody taking care of them. That's got to be throughout their entire kind of species. But also we've talked on the show about different species recognizing each other as well. So there's definitely an advantage to knowing your neighbors.
Who are the fish in your neighborhood? And the zookeepers in your neighborhood? And the really nice people who feed you peanuts? All right. It's figuring out being an animal in an environment with all the stimuli.
Hey, that's us.
Who do you know? Who do you not know? Who's dangerous? Who's not dangerous? What are the things that are safe and not safe? Yeah.
Is that a familiar dog you see down the street? Or have you not seen it before?
Oh, I know that dog. It's a pit bull. You stay away from the mean pit bull down the street.
It can be sweet too.
I know. Yeah, I grew up around people who trained their dogs to fight. So I have
Stockton. What are you going to do?
Yeah, exactly. Come on.
It's sort of the whole story there about watching out for predators. I remember this from there was a fish tank in high school from the forestry class, and somebody had put in too many minnows. There was a trout or something in this giant fish tank, and the teacher warned us not to put too many in because the trout would normalize with them and stop eating them. They're feeder fish that you could put in to feed the trout. But if you put too many in, it would no longer be other. It would no longer be food, and it would stop eating them. So even a predator can can become part of a community of what was the food fish if it hangs out there long enough and is surrounded by them. Yeah. They become the familiar, and these are not my fish friends. Also, why would I eat them? That doesn't make sense. They're helping me watch out.
That's Gary and Fred and Joanne. I'm not going to eat them.
Don't eat my fish friends. Those are my fish friends. Well, this is This Week in Science, and I hope that we are your science friends, bringing you science every single week. Justin, what science do you have right now for everyone?
Yes. An exceedingly rare butterfly species was discovered in San Francisco.
It was?
Yeah. Mm-hmm. This is exactly the sort of place you would expect to find a species of butterfly that no one had noticed before. There's a city overfilling to the brim. There's no parking spots left in San Francisco. Don't bother going. Too many cars already.
Is it the Mission Blue butterfly? Because I know that one's only found in San Francisco.
It's the Xerces, Xerces, Xerces blue butterfly. It was actually last seen flapping its iridescent periwinkle wings in San Francisco in the early 40s. And it's generally accepted that it is extinct. Yep. They think it may even be the first American insect species destroyed by urban development? That can't possibly be true, the first one. Maybe it is. But there were some questions about whether or not it was actually a species to begin with or a subpopulation, possibly of the butterfly that you're describing. This is in Biology Letters. Researchers analyzed the DNA of a 93-year-old Xerxes blue specimen in a museum collection, and they found its DNA is unique enough to merit being considered a species. And that is the rest of the story.
So it was found, but it's dead.
Oh, yeah, it's been extinct for 50 years.
Right. So this is a posthumous species designation. Discovery.
Well, I should have put a comma between discovered and
dead.
50 years new species it turned out to be. But
it's an old dead species that used to be unique and used to be amazing. Congratulations, humans. We killed it. Good job. Oh, jeez.
I wonder why nobody talks about, like, is that something we could bring back? Could we use DNA to bring back an extinct butterfly to San Francisco? I think we could get people behind that. If you're not behind mammoths or Neanderthals, which is probably a little dicier.
How about
maybe
something
a butterfly?
that's only been gone 50 years?
Yeah.
Huh?
See, that would be okay.
That would be okay. All right, we found one. We found one. All right. So now we need to get some funding and get this butterfly back. The next story is more things change. The more bacteria on the ocean floor don't care. Winter, summer, spring, and fall. Even on really windy days, the bacteria on the sand on the floor of the ocean is always inhabited by the same bacteria. There wasn't an intuitive find. One of the things about water is that the makeup of bacteria floating around in water changes seasonally. It is affected if there's 24-hour sun or 24-hour darkness. They change if it's getting colder waters versus if it's warmer waters. But the sand on the ocean floor, even though different regions might have their own distinct populations, or different regions of ocean floor might be distinct from each other to some degree, the populations there don't change. Once that is established, it stays the same despite seasonality, light, temperature, any of it. And it's sort of like something we've talked about before, about life not showing up on this planet many, many different times. Talking about all the niches being filled. This is literally all the niches in a grain of sand being filled by the same bacteria. And having simply no room for an invader to take up enough territory or real estate to get a foothold in the ocean floor. This is all research out of marine microbiology in Bremen, Max Planck Institute for Marine Microbiology in Bremen, Germany. They're describing this in the journal ISME Communications. What they found was for them very unintuitive, having studied bacteria in the waters, ocean waters. But yeah, there is absolutely nowhere else. And it's also is working as a pretty huge filter. The bacteria that are there are breaking down chemical components that are in the seawater, and they're creating a sort of filtration system of different compounds that then goes deep into the sands of the ocean floor and may not ever come back up again.
So they fall to the bottom and they never come back.
But it's an area, the area they were studying, that you were showing there, does get that midnight sun, does get that 24-hour-ish, near 24-hour sunlight at some points, where the sun is still in the sky well past midnight. Didn't affect the bottom of the sea, didn't care. Didn't care.
So is it the grain size of the sand? Is it the light availability? Like they didn't care about the light. They're at the bottom of the sea. It's dark. But
well, and I don't know. So that's a great question because I think what they're not like this where they're studying doesn't look like deep ocean. So there's probably got to be some light filtering through there. But the point is that the niches are so filled. They're so homogeneous at this point that there isn't room for a whole lot of infiltration by another species to even get the foothold. And they even went to the point where they're looking at sand that is facing down versus facing up in their analysis. The sand that's facing down is completely covered in every little niche. It's just like, and then the exposed sides, because I guess the play of the water or something, have a much lighter inhabitants there. Well, so if the only thing that is available is the thing that's hard to hold on to, also, that sort of adds to the inability for other bacteria to then infiltrate. And it sounds like it's just a really healthy, happy environment that's providing everything that the bacteria need, that they can remain so stable, despite any seasonality changes and that sort of thing. But it just really reminded me of our conversations about how come evolution hasn't shown up multiple times.
Right.
When you get down to the point where you have every
room.
Every nook on a grain of sand is occupied. There's really nowhere for a next thing to move into.
No more angels can dance on the head of this pin. No more. It's done.
Exactly.
No more room. Get off my pin. Get off my grain of sand. Oh, that's fantastic. Yeah, I think that's just a fascinating insight that just at a certain point, the bacteria, the life there, it's efficient. It's taking all the resources. There's no room. There's just not a place for more inhabitants. And yeah, I wonder if it's the same everywhere or just where they looked.
Yeah, that's a great question. So of course, there's always more research that needs to be done. Always. One of the things that they did, that you mentioned, is they did want to look at if there is any sort of seasonality change in enzyme expression. Maybe diet does change. Maybe there are different metabolic activities taking place still seasonally within that same population. So to that point, they may be more plastic in their ability to change strategies. That could be something that's at play. Why they don't have to abandon an area when there is some temperature change and that sort of thing, where conditions are different than you would normally expect them to be. So yeah, that's still on the table. It's like, how are they so good at maintaining a stable population?
I hope we will find these things out in the years to come as researchers study these questions more and tell us more about our universe, about our planet. I have some science stories for you. You want to talk about mockingbird song? Some of you might not consider it song. That repeating, ew, 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 when the mockingbird sits outside your window early in the morning before the sun has even risen, copying the sounds of the car alarms. Mockingbirds.
Fascinating birds.
Yes, they're fascinating. Blair, I hear what you're doing.
I'm done. That was the last one.
I hear you. But a group of researchers got together and they used their various strengths to investigate mockingbird song and to explore the various structures that mockingbirds use in their song. And while you may, in your annoyance of the morning mockingbird, not think of it so much as a song, mockingbirds are amazing mimics. They are able to mock many species in the animal kingdom. Their mimicking, though, does have limitations. According to an article from Ars Technica, the birds can mimic blue jays, but not ravens, tree frogs, but not bullfrogs. And so they have a repertoire of particular species and sounds that they are able to mimic, but not others. So researchers have studied how they sing their songs, and we know that mockingbirds repeat each syllable 3 to 5 times, and these repeats are separated by little breaths, and then they'll have a new syllable that comes in. And 25% of the syllables that they use don't appear very often, but they can have thousands of phrases individual birds can put so many things together. So how do they choose all of these different sounds to put together? And the researchers looked at different stylings, what they called morphing modes, and morphing modes are what they call like a variation on a theme. And they recorded a group of 5 different mockingbirds, 3 in the spring, and then a couple of others from a birdsong database. And the researchers, there was a researcher who's good at statistical analysis of animal signals. So that researcher was really good at putting the statistical analysis together. Another researcher is a music philosopher who studies connections between music and nature. And the third, a field biologist who studies the songs of mockingbirds. And this particular quote from the study, again from the Ars Technica article, says, "When confronted with complex mockingbird song, a musician will hear one thing, an ornithologist another, and a signal analyst something else. The most complete human knowledge of any natural phenomenon comes from combining distinct human forms of knowing. No one perspective negates the others. They are strongest when applied together." So I love just their philosophy of how they came together to use their strengths to analyze mockingbird song. So anyway, they looked at all these different spectrograms of the songs and the morphing modes of how the strategies were being employed by the mockingbirds, and they came up with 4 different strategies. There's a timbre change, which is like the difference between a flute and a piccolo, or a flute and a clarinet. You know, that you might have a B-flat instrument, but because it's a different build, a different construction, it has a different timbre. There's also pitch change, which is actually changing the pitch, which I guess that would be more appropriately the flute to the piccolo. Stretching the transition, so taking an element of the song and making it longer and longer, or squeezing the transition, like taking an element of the song and smushing it up and making it faster and faster and faster. And they figured out how the birds do all of this, but they say that every transition involves a mixture of more than one of these modes. And they use this for their testable hypothesis, and basically ended up determining that the way that mockingbirds put their songs together is very similar to how humans put their songs together. The timbre morphing mode of the mockingbirds, everything about the syllable stays the same except for the timbre, and the mockingbird shifts from the mimicking call of one bird species to another with a different tonal quality without changing the rhythm. So they will go from the song of a non-mimetic bird to a mimic of a blue jay pump handle, but it's the same sound, just a different timbre. So they'll switch to something that sounds very similar in how they're singing it. And this is something that is seen very often in Tuvan throat singing, in actuality, where they'll have the similar sound, but then different timbre. The birds then will also use different pitch changes. So they'll do a long flicker call and then change the pitch to their own call, but it's very similar to the flicker call in the beginning. And we see that happen in music, like in Beethoven's symphony. And then there's also evidence of the lengthening or the slowing down of a transition. We've seen that in Disney in some of their music. And additionally, the clumping up and shortening of phrases is something that has been seen in other music as well. So there are many examples. I think Kendrick Lamar uses a lot of these examples as well. And the paper actually links to Kendrick Lamar as an example of a human musician that mockingbirds sing similarly to.
I want to know if our brains are activating in the same place as theirs.
I think that's a really interesting question. I have no idea. Yeah. The question of, if they, as they're making their transitions and as they're making their choices for their song elements, it's like, oh, I'm singing, and then I'm going to do, and do an element that sounds exactly the same, but is at a different pitch? Or am I going to have something that has the same rhythm, but the timbre is different? Or, how am I going to make this musical shift? But along the line of like things that animals do that are interesting and the abilities that animals have is the big question of whether or not the birds are doing it because they think it sounds good, because they have this vast repertoire to choose from. Are they making these choices from some sort of judging of good composition versus bad composition? What sounds good? And I guess there is some evidence that some species of birds do have like a qualitative sense of what their songs sound like musically that they try to do.
Still doesn't explain the car alarm.
Aesthetics, right? Yeah.
It
doesn't explain the car alarm.
It doesn't explain the car alarm. But yeah, so I want to know what's going on in their brain versus our brain when that music is being composed, and similarly when it's being heard. Because I'm assuming that ears across the animal kingdom have similarities, and that therefore what we hear from sounds is similar to other animals. At least to terrestrial animals, I will say. Yeah. Animals on dry land. So if I make that assumption, would, to your point, the same sounds be considered pleasurable across species and desirable? Yeah. And then, so yes, this is what I want to know. Like, what was the first animal to sing? And evolutionarily,
and did they like the way they sound?
Yes, or is this something that happened through convergent evolution, that even possibly that humans heard birds and then kind of stole it, right? These are, yes, I'm very curious about all this.
We do know that there are brain similarities, and we use bird song as a model, a neurological model for human speech. And there are a lot of neurological studies that look at bird song, bird brains versus human speech production and human brains. And there are many similarities. And so I think that particular question of what is lighting up and what is getting activated as the choices are being made, and if there's similarity there too, is that,
But then what I want to do is I want to get the artificial bird ear. To sort of translate it into what they can hear from another bird song, or because they're going to be getting so much more nuance. It's like how we only just discovered mice can sing. Mice knew it all along. Mice were listening to mouse songs and like having fun, but we only just picked it up because the range
I hear?
Like the whole world of recording and then putting into our range animal noise. Right. We should put out a CD of just animal sounds.
Yes.
Like, I would love to hear that.
Well, we should put a CD of animal sounds where each track is followed by us trying to replicate that sound. Really, that's what it should be.
That sounds like a lot more fun, actually.
Yeah, that would be fun. Last study of the night, researchers at UC Berkeley looked at empathy in rats. And we know that rats are empathetic to other rats that they know. So like the fish, they have their familiars. They have their in-group, they have their out-group, and they will save a rat that is one of their in-group over one of the out-groups. So these researchers, they published their study in eLife, wanted to know what, beyond just that empathetic aspect and the group behavior of that, of saving someone you know versus not saving someone you don't know. They wanted to know how the brain of the rats was responding to the situation. And what they found is that there is a brain network involved in this empathetic helping, very similar to humans. They used fiber photometry, immunohistochemistry, calcium imaging, and used other tools to look at the experience of empathy in response to other rats' distress. And so the brain of the rats. They discovered that to act on the empathy, So no matter whether it was in-group or out-group, the empathy network was stimulated, whether the animal they were looking at who was in distress was in-group or out-group. But in order to save the animal in distress, their reward circuitry needed to be stimulated as well. And they think this is something that happens as well in humans. And the big insight from this is that we need to broaden our sense of who is in our group, who is someone we know, who is someone like us. We need to broaden that so that more people, or rats, are included in not just our experience of empathy, but also within our reward circuitry so that we want to help more people. And
or the sci-fi version of that would be to give somebody an empathy chip, right?
Yeah, sure, you could give somebody an empathy chip, but it's not the empathy chip, it's the reward chip. It's because regardless, you can look at anybody and you're going to feel empathy. But it's whether or not you act on that empathy is the big point. It's, do you give spare change to the homeless person on the street?
Mm-hmm.
Who has change? One. like, that's, I mean, yes, back when paper, coin, metal money was a thing. That was the thing you could do.
But beyond that, it's like, how do you decide who to give, your charity to? Mm-hmm. How do you decide who to help when they've fallen down? Who do you decide to help when their house has burned down? Who do you decide to help when an oil pipeline is being proposed? You know, these are the big questions. Who's in our group? Who's not in our group? And it's just really interesting to see that very, very specifically in these rats, the reward pathway needed to be stimulated. It needed to be triggered. And it only happened with rats that were in group. And so if we can expand our idea of in group.
Yeah. And so this
more reward, more dopamine, people.
This connects really well to a study, one of my favorite studies we've ever done on the show, where they had rats that were related and they had one trapped, and then they had to see if they would help the other one out, right? Then they had rats that weren't related and they didn't help. And then they had rats like a black and white spotted rat that was raised with a bunch of all white rats. And that rat was let out because he was raised around them. And so he was part of their social group, right? And so I see this tied into that, in that, how do you develop empathy for all people? And exposure is the answer. Yes. You have to have people in your society, in your neighborhood, in your culture, in your area that is in a relevancy to you to represent togetherness and part of your group, I guess.
Yeah. And it is Robin Williams' birthday today, I believe. And something he said once upon a time, testifying in front of Congress to try and get Congress to solve the homelessness problem, said, you don't help people by lifting them up. You help them by keeping them from falling down. So if we can accept more people, broaden our definition of our neighborhoods and who exists in those neighborhoods, maybe we can keep more people from falling down in the first place.
Yeah.
Thanks, Robin Williams. Mm-hmm. Yeah. Anybody else have more stories? No? All right. I think we have come to the end of another episode of This Week in Science. We did it. We brought you the science. It wasn't all the science, but it was a bunch of good science. And
it's a good sample. Smattering.
Yeah, smattering. Lots of poop. Lots of animals.
That's a scattering.
We covered the whole ocean floor.
We covered the ocean floor. We went from mountaintops to
planet.
Ocean floors. Yes. I think we gave it a good tour this week, everyone. Thank you for listening. I do hope that you enjoyed the show. Shout outs now to Fada, thank you for your help on show notes and social media. Gord, thank you for manning the chat room. Our other moderators, thank you so much for all your moderation. Identity4, thank you for recording the show. Rachel, thank you for your editing and assistance. And I would like to thank our generous Patreon sponsors. Thank you to Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Kira, Carl Kornfeld, Jen Myronik, Melanie Stegman, DeKramsta, Karen Tazi, Woodaya MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Parangor of Sharma, Shoe Brew, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavis, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Atkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you want to support us on Patreon, head over to twist.org and click the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific Time, live, broadcasting from our YouTube, Facebook channels, and from twist.org/live.
Do you want to listen to us as a podcast? To build anticipation, just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well. And remember, it's important to establish who your friends are, as we discussed today.
Yes. For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org. And you can also sign up for our newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS in the subject line, or your email will be spam filtered into the depths of the ocean floor amongst many types of bacteria.
It'll just get lost down there.
Yeah. No room. Yeah, you have to sleep in the sea.
Yeah.
If you want to, you can hit us up on the Twitter where we are at Twist Science, at Dr. Kiki, at Jacksonfly, at Blair's Menagerie. We love your feedback. If you'd like us to cover a topic, or you have a suggestion for an interview, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.